Module 8, Objective 1, describe the sensory organs of smell and trace the olfactory pathways to their destinations in the brain, and explain the physiological basis of olfactory discrimination. Our sense of smell is seated within the nasal cavity. We have several olfactory organs within this nasal cavity, and ultimately these organs comprise two major layers, the olfactory epithelium, which is indicated here in the purple, and then we have the lamina propria. Remember, the lamina propria is the connective tissue that lines or supports any mucous membrane. So within the olfactory epithelium, situated within the nasal cavity, we're going to have that lamina propria supporting it, but also nervous components, which we can see here. The nervous components include the olfactory nerve fibers. These fibers are going to penetrate the cribriform plate of the ethmoid bone and innervate the olfactory epithelium. They synapse at the olfactory bulb, and the olfactory bulb is situated on either side of the crista galli of the sphenoid bone, the ethmoid bone rather, and they sit on top of that cribriform plate. And then the olfactory tract is going to carry those sensory information from the olfactory bulb back to the central nervous system. In terms of the olfactory epithelium, we have several cell types. We're going to have olfactory receptors. These are going to be modified neurons here in blue. And you can see that those receptors are going to send their little hair-like fibers down through the cribriform plate and innervate the nasal epithelium. In addition, we have supporting cells, which help support those neurons, and then a stem cell to replace any worn-out olfactory sensory neurons. The lamina propria, identified right here, again is that connective tissue associated with mucous membranes, and they're going to bring in all of the vasculature, the nerves, and it's comprised largely of aureliar tissue. Reception begins when an odorant molecule binds a G protein coupled receptor, and in doing so it's going to generate a graded potential and a local depolarization. We call that potential the generator potential. Of the afferent fibers that leave the olfactory epithelium, they collect in bundles of 20 or more fibers. They're going to penetrate the cribriform plate of the ethmoid and then synapse at the olfactory bulbs. From the olfactory bulbs, you get sensory information traveling along the olfactory tract to the olfactory cortex, hypothalamus, and limbic system. The olfactory information is the only sensory information to actually reach the cerebral cortex directly. All the others tend to be funneled through the thalamus. So the thalamus has some say into what gets projected onto the cerebral cortex, but olfaction goes straight to the cortex. This image is a little complicated, but it sort of explains the role of that G protein. So we start off with the binding of the odorant molecules onto the receptor proteins, and that's going to lead to an activation of an enzyme called adenylate cyclase. And adenylate cyclase converts ATP into a second messenger molecule called cyclic AMP, or CAMP. And it's the CAMP that ultimately binds sodium channels, causing that to open. Sodium then can flood into the cell and generate the graded potential. If that graded potential is strong enough, an action potential will be generated, and that's going to relay that sensory information to the central nervous system. In all, we can distinguish thousands of chemical stimuli. In comparison, dogs have 72 times more olfactory receptor surface area than humans, so their sense of smell is about 10,000 times better than ours. As we age, our olfactory receptors die, they decline, and they're quickly replaced by those stem cells. But in all, as one ages, they're going to lose the total number of neurons. So as we age, our sense of smell tends to decline.